Hello everyone! Today we are going to explain some concepts about cancer, especially anti-cancer drugs. This topic, anti-cancer drugs, will be discussed in two parts. The first part is, as we always do, an introduction we need about pathophysiology, general principles, and the language of this new world we are about to venture. Because you can never understand anything about anti-cancer drugs or its mechanism of action or even the language spoken unless you understand the general principles and other concepts we are about to illustrate. Now, we are going to discuss only the therapy. All bases we need to know so you can understand the next video at ease. Some of these bases are suitable for undergraduate level, others are for postgraduate level. Anyway, we can but explain these main topics. So, let's firstly begin with it. Understand it, and hopefully, you will find it easy and straightforward. We have some titles to begin with, like what does cancer mean? What distinguishes cancer cells from other cells? What happens to normal cells so that they transform to cancer cells? And what problems we may experience while we are trying to treat those cells? In other words, what are the hallmarks of cancer? Number 2 are some terms you may face while you are studying pathology, such as proliferation, differentiation, and undifferentiation. We need to get familiar with these terms to understand how drugs work. Number 3 is one of the fundamental topics regarding basic medicine, and this topic has a close relation to cancer, so we need to comprehend it very well. Number 4 are some ideas about genes and gene rules in cancer. Number 5, we are going to have a glimpse about major topic called cell signaling network, or what is known as cell signaling pathways. Number 6, we will have some information about growth factors, their relation to cancer, and how can they lead to uncontrolled proliferation. Number 7 are new topics such as telomere and telomerase. Lastly, number 8 is going to be treatment options. What choices are available for cancer treatment? All of these topics can be long chapters in huge books about cancer. However, we will try to summarize these long chapters in one page in front of us on this screen, where we will have main concepts that you cannot graduate without understanding. Because without it, you can't study cancer thoroughly. Let's go step by step and start with the first topic, which is hallmarks of cancer, or what distinguish cancer from other diseases. We was told before that any cell in our body, any cell whatsoever, can transform under certain conditions into cancer cell, from normal cell into cancerous cell. This process is called malignant transformation. What are the reasons behind transformation of normal cell into crazy, unrational cancer cell? There are so many reasons, however, they are of little significance regarding our topic. If you wish, you can always read about it in pathology books or any other source, because there are so many reasons, but this is where we start. Cell has already transformed into cancer cell. What distinguishes this cancer cell so we can imagine how we may intervene with a drug and what problems can we face? Number 1. The first quality cancer cell acquire, which is uncontrolled proliferation. Proliferation means cell division, so cancer cell go crazy and keeps dividing all day without stopping. You can use proliferation or cell division because both terms are synonyms. However, in cancer language, it is preferred to use proliferation rather than using cell division. This is the first hallmark or the first problem facing us. Cells have uncontrolled division and we can't regulate them. Number 2 is resistance to apoptosis. Cancer cells are characterized by their strong resistance to what is known as programmed cell death. When normal cells confront an insult, they die easily through activation of apoptotic machinery program. However, cancer cells are very resistant, so they require violent intervention in order to stop. Number 3 is induction of angiogenesis. Angiogenesis means blood vessels. Cancer cells have the capability of releasing something we are going to talk about in minutes, called growth factors, such as vascular endothelial growth factor. This vascular endothelial growth factor enables cancer cells to build new blood vessels. Because without these blood vessels, cancer can't form colony nor form a tumor. These cells are always dividing, forming colonies and large tumor, so they need feeding. And they have their maneuver to form their own blood vessels in contrast to normal cells. Number 4, or the fourth quality, is invasion or metastasis, and they are something to be considered. They are qualities we fear most about cancer. When normal cells divide for any reason, and soon as normal cells finds another neighboring cells, a process called contact inhibition occurs to stop the dividing normal cell from invading other neighboring cells or tissue. Because cancer cells are in continuous search for blood vessels, so they can permeate through it and follow the bloodstream anywhere. That is how it can form colony anywhere and tumor spread in different parts. This is called metastasis or secondaries. This is a very aggressive quality about cancer. And that is what we fear most about cancer. Because as long as cancer is localized, there is always hope for surgery. However, once you hear cancer begins to enter bloodstream and metastasize, here and there, it's said that patient has entered stage 4, as pathologists say, and now treatment is only palliative and there is no hope for recovery. That's why both of them, either invasion or metastasis, both are such terrible qualities of cancer cells. Number 5 is a new dazzling topic, which is avoidance of immune destruction. Cancer cells developed strategies to disappear and deceive immune system. It is supposed that cells of immune system, we have many cells in immune system capable of detection of any abnormality, so they can localize any abnormal behavior and eliminate it. However, cancer cells developed many techniques enabling them to hide from immune system and evades immune response. Subsequently, immune system stands still and watch as cancer cells grow and form large tumors. For a long period of time, scientists have been occupied by this topic because they wish to understand the techniques enabling cancer cells to deceive immune system. Now, you may have heard of social propaganda about new drug that can completely cure cancer. Anyway, this new agent that we are going to discuss in drugs chapter acts through only one mechanism, which is to reveal cancer cell to immune system by counteracting those strategies cancer cell use to hide from immune system. This is what pharmacologists do. They find the problem, then they design the drug. Lastly, or the last quality distinguishes cancer cell is loss of function. Cancer cell is a cell without purpose. Its only function is to wander the body, destroying adjacent tissue and destroying its owner, because it won't die unless it kills its owner. This whole part about hallmarks of cancer is explained only for one reason: to tell you we as pharmacologists are always looking for the problem and how can we intervene with a drug. For example, the first quality, uncontrolled proliferation. Cancer cells are growing and dividing so much quicker than normal cells. This point of difference enables us to target it with a drug that can attack those rapidly growing cells. Another one is induction of angiogenesis, which enables us to invent a drug can deprive cancer cells from such quality, preventing them from forming new blood vessels. Subsequently, cancer cells can't grow and die. We can also target immune system and so on. Now, let's shift to the second topic, which is proliferation, differentiation, and undifferentiation. These terms may be closer to science of pathology than pharmacology. However, we need to gather even little pieces of information which we need or you may find useful in many other areas. This is normal cell. Then, for some reason, it transformed into cancer cell. This cancer cell, as we now know, has acquired the quality of rapid proliferation or rapid cell division. So, this cell will divide into 2, then 4, then 8, and so on. Those 8 become 16, 32 until they form a huge tumor. This doubling process from 2 to 4 to 8 and so on takes time. And this time taken as cell divides is called doubling time, which is time taken by the cells in order to double its number. This term, doubling time, is somehow specific to cancer studies. All cancer cells have certain doubling time. You may find this time has a scale from 24-hour in cases of cancer type called Burkitt lymphoma. In this lymphoma, or lymphoma cells are doubling each 24 hours. So it has a very rapid progression. Sometimes this time scale can extend to weeks or months, like what happens in breast cancer. Mammary cancer has doubling time of 3 months, 90 days, every 3 months. So the less doubling time is, the more quickly tumors can progress. Again, shorter doubling time means rapidly progressive or highly malignant tumor. And when doubling time is longer, it gives us opportunity to deal with it, and so this cancer is somehow less aggressive. This is the first concept we need to understand from this photo. Second one is differentiation and undifferentiation. Allow me to draw your attention to this one. This cell is normal. This cell here is normal cell. Once it changes into cancer cell, during the first levels of transformation, cancer cell still shares some similarity to its progenitor or mother cell. So, when pathologists are looking at some samples under microscope and can still recognize its original tissue, such as breast cell or prostate cell, because it still retains its shape. They say these cells are differentiated, meaning I can still recognize it with my eye and assign it to its original tissue. However, with each cycle of cell division, you can observe that deformity in the shape of the cells increases and it loses its similarity to its mother cell. So, you find these cells are not similar to this cell. With each cell division, the resulting colony is less similar to the mother cell. until you reach a stage like this where cells lose any similarity to the mother cell, so that pathologists seeing it under microscope can no longer distinguish the original tissue or the source of this tumor. In other words, those cells have lost their identity. They have lost their shape. No one can distinguish them because they have become undifferentiated. This is the second concept we need to learn from this photo. Now I want to draw your attention to something important in this concept. You should know that with each cycle of cell division, cancer cells become more aggressive and resistant to drugs. If a patient comes in the first stages and pathologists say the cancer cells are differentiated, this cancer can respond to drugs. However, with more time, when the patient reaches this late stages, cancer cells become undifferentiated. This is very late stage in the disease progression, and now cells have become more resistant to treatment. Now for the major topic we are going to discuss, and I need you to understand this topic thoroughly because it's one of the fundamental topics that every medicine student should be aware of. Either you are a fresh medical student or a Nobel Prize winner in medicine, you must understand the cell cycle. Let's assume that our body cells under normal conditions without need to divide or do anything, our cells are somehow sleeping and they are in what is known as resting state. Let's assume that my skeletal muscles right now are in resting state without any need to divide. My skeletal muscles are somehow sleeping. They don't have to work or have to divide. They are resting right now. So, in this situation, growth of these cells is 0 or takes number 0 because there is no division. So the cells are now in G0 or growth 0. They are resting cells. But if these cells, for any reason, are ordered to divide, of course, we are talking about normal cells. If they were ordered to divide, this order in molecular biology is termed mitogenic signal. Now, the term mitogenic signal means orders to divide. So, once cells receive this mitogenic signals, they start to enter a process known as cell cycle. First of all, this resting cell enters the first phase termed G1 or growth 1. G means growth. What happens in growth 1 or G1? Not so much actually. This cell only increases in size in preparation for cell division. It is ordered to divide, so it must prepare by increasing its size as if it were pregnant. If this phase is completed, then the cell enters the next phase, which is so important and termed S phase. S is short for synthetic phase. It is called so because in this phase, cells double their genetic material or DNA. The 46 chromosomes are cloned and doubled. If this phase is completed safely without any complications, then the cell enters the next phase or the third phase, which is termed G2 or growth 2, and it means that the cell increases its size furthermore. Here it is termed G or growth because it increases in size. And here cell also increase its size furthermore. But why? Because it has copied its DNA material. However, we still need to copy other cell constituents. We still need to copy some of the mitochondria. We still need to copy other components of the cytoplasm. Because this cell is supposed to divide into two daughter cells, so we need to copy other components present in the cytoplasm. If this phase is completed without any problems, then we enter to the last phase. Now that everything is ready, cell enters this phase colored with purple and termed M phase. M is short for mitosis. As if every phase takes the first letter from the name. G is short for growth, S is short for synthesis, and M is short for mitosis. Now we are in the serious phase of mitosis, and the first thing this big cell does is to form this green body at the two poles of the cell. This green body is called centrosome. Why does the cell forms centrosome? Because the centrosome is about to extend some filaments termed mitotic spindles. This is a very important term we are going to use more often. Some people may call it microtubules, others may call it microtubular system. So, what are these mitotic spindles or microtubular system? These mitotic spindles are filaments from both centrosomes at the poles. These filaments arrange chromosomes in the equatorial plane of the cell. Now we have 46 chromosomes with their new copies, so we need to arrange them in order to proceed smoothly. These filaments, termed mitotic spindles, are named after spindle threads because they arrange chromosomes in the equatorial plane, and this stage is termed metaphase. Once chromosomes have been arranged in the equatorial plane, mitotic spindles pull each set of chromosomes towards themselves. All of this occurs in order to reach the next phase termed telophase or cytokinesis, where cell actually divides. Constriction occurs in the middle of the cell, so it divides into two identical daughter cells. Both are identical to the mother cell. This first stage is termed prophase, which is just some preparations. Then the next stage is metaphase, when mitotic spindles are formed. The third stage is termed anaphase, when chromosomes are pulled away. Lastly, we have separation stage that's termed telophase or cell cytokinesis. Now we have finished cell mitosis, so we need to look at these cells. Either they are parked in the garage or enter G0, so they can have some rest now. Or they may receive other mitogenic signals and continue dividing for 2 or 3 cycles or as many times as the body demands. So for example, there are some cells known to be dividing a lot like bone marrow cells. Other cells don't divide much like heart cells and brain cells and so on. This is a brief illustration for events occurring during cell cycle. Now, here are some points worthy of Nobel Prize. If you intend to get one, you need to focus on those small points. Here I have 4 red points, which I didn't tell you about before. These red dots are termed checkpoints. These checkpoints are like security checkpoints you encounter at airports. If you travel from one country to another, you may encounter more than one checkpoint. This is exactly what happens in these red dots called checkpoints. 3 of them are very important, and the 4th one is not as much. Even some authors consider them to be only 3 checkpoints during cell cycle. What is the role of these checkpoints? These red dots are for monitoring of cell cycle. Was each step completed successfully or did something go wrong? For example, in G1, now the cell has completed G1. This checkpoint looks for any abnormality in the cell after completing this phase. Was cell growth completed successfully or did something go wrong? If everything was proceeding smoothly, cell enters S phase. After completing S phase, another checkpoint sets in to check for the process of DNA doubling. Were all chromosomes doubled correctly? Were the 46 chromosomes copied completely, or did one went missing or translocated? Or if some chromosome is missing an arm? Now, if everything is in place, cell is allowed to enter the next phase called G2. After we complete G2, same scenario happen once again, where checkpoint number 3 perform one last check on the cell. Here, after G2, because cell after this phase is entering actual cell division phase where it cannot go back. So if something wrong occurred and we missed it, these daughter cells are produced carrying an error. Indeed, as you think, this error can lead to anything like a tumor or cancer. What are these checkpoints? Or who is the one responsible for this security check? Although this concept seems easy enough, some scientists won their Nobel Prize studying it. This checkpoint is made by very complicated and evolved proteins. These proteins are called cell cycle regulators. Group of proteins that interact with each other in order to monitor cell cycle. Cell cycle regulators, or this group of proteins, are classified by scientists into two large groups, positive regulators and negative regulators. What are positive regulators? They are a group of proteins helping the cell to proliferate, convincing the cell to divide and run this cycle. Who help the cell making her decision of division? They are those gentlemen called positive regulators. I'll mention some examples for you, surely not all of them, because their numbers are unknown. However, some of the most important proteins here are family called cyclins and cyclin-dependent kinases, or CDK. This family was firstly discovered in 2001. As I told you before, such discoveries are made by individuals or group of people after dedicating so many years studying them. Cyclins are one of the most important proteins responsible for turning on the cell cycle, maintaining it. That's why it's termed positive regulator. Second group are growth factors, which will be discussed in detail soon. We have so many types of growth factors, like epidermal growth factors, platelet-derived growth factors, and so on. They are such an important family, and every day there is something new to it. Third family are signaling proteins or signaling molecules, such as ROS, RAF, ERK, AMAP, and so on. These are few examples because they can be 400 or 500. Every day there is something new to it. Now these are some examples of proteins called positive regulators, and positive regulators mean it has to turn cell cycle on and help the cell to reach cell division. On the other hand, we have some brakes, meaning they are responsible for stopping the cell cycle, such as a very important protein don't you ever forget about called P53 protein. Another are BRK1 and BRK2, short for breast cancer 1 and 2. There are more to it, however, we are mentioning only a few examples, so you can comprehend how everything works. This positive regulator function is to move cell cycle in this direction, forward, on condition that they have performed the security scan after each phase and found nothing wrong, so they allow the cell to continue its cycle. However, once these proteins find out an error at any stage, then this protein called P53, the strongest protein among them all, intervene. Once it detects something went wrong, it immediately stops the dividing cell from continuing this abnormal division that may result in formation of cancerous cell. If the error occurs here in G1, then P35 stop it here, preventing it from entering the next phase. Then it turned on a machinery called apoptotic machinery, which is the machinery committing this malformed cell to suicide because it has an error that must be stopped. This error must be stopped at any cost. Thus, the malformed cell can't be allowed to live. If G1 is successful but an error occurs here during duplication of DNA, P53 stops the cell here, preventing it from entering to the next phase, turning on a apoptotic program in order to eliminate this threat and so on. So there is balance between regulators moving the cell cycle and regulators stopping it. This balance is like balance present in cars you drive. Any car has two pedals, gas pedal increasing car speed and brake pedal stopping the car. This is exactly what happens with these two groups. This are gas group, moving the cell cycle, and this group is brake group, stopping and killing the cell if any error occurs. Now you understand this concept, but before we go to the next slide, there are some points I still want to clarify. I've been talking with you about protein families and their function, so if I may ask you, where did these proteins come from? What synthesizes such proteins? Proteins are synthesized by genes. That's true. But you should know that proteins are mostly named after genes. For example, this P53 I am talking about is cell cycle brakes. Gene responsible for P53 protein synthesis is also called P53 gene. Then how can we differentiate between them both? So if you are reading some books about cancer and find this P53, how do you know if the author means P53 protein or P53 gene? If you saw this P53 written in ordinary font style, then the author is talking about the protein itself. However, if you saw it written in italic like this, then the author is talking about the gene responsible for protein synthesis. Always remember that proteins take their names after genes responsible for their synthesis. They have the same name, 3 or 4 letters, sometimes are alphanumeric characters. So if you saw the name written in ordinary straight font, then it's about the protein itself. But if you saw the name written in Sloopin italic font, then it's about the gene. This is the first note. Second one is, if there is a mutation in any gene responsible for synthesis of these proteins I mentioned, or any other proteins participating in cell cycle, if any mutation occurs here or there, then you lose control over cell cycle. In this case, the body becomes vulnerable to uncontrolled division of cells that can turn into a tumor. For example, if a mutation occurs in genes responsible for synthesis of these cyclins, or if there is any mutation in one of these growth factors, or even mutation in the receptor of growth factors, what occurs in these cases? These proteins are positive regulators, which means they are proteins responsible for moving or turning on the cell cycle. So the result is, this mutated protein drives the cell into a state of continuous proliferation without resting or parking in the garage. 1, 2, 3, 4 cycles and so on, continuous cycles without stop. Proliferation machinery doesn't stop. Why? Because there is an error here. And now this is the rule of these negative regulators. If they spot this error, they stop the cycle immediately. But what happens if this patient also has a mutation in this group? For example, another mutation occurs in the P53 gene or in breast cancer 1 or 2. In this situation, brakes is also absent. So on one hand, proliferation machinery is continuously working without any inhibition, and on the other hand, car brakes is also absent, and there is no way to stop this speeding car. In such situation, it is inevitable that a tumor sits in here. It is enough to say that any mutation in P53 gene is responsible for 50% of all of these tumors worldwide. So if you examine all cancer patients in the world and make a statistic, you find that 50% of them have mutation. In the brakes, car brakes are absent, and 30% of them have mutation in ROS gene, which is gas pedal of the car. So here is a mutation responsible for 30% of all cancers, and P53 mutation is responsible for 50% of all cancers. This is the base you need to know in order to understand the following slide. Here we will talk about cancer genetics. In the past, we used to make some questions for brilliant students, or when we had a group of students and we wanted to see how far those students understood molecular biology. We used to ask them about cancer genetics. We only wanted students to say these organized points. By the way, these points are the same explained in the previous slide. However, I'm going to say them in a more organized fashion. Again, we have two sets of genes, two large groups in our cells. The first one that are like car brakes and termed tumor suppressor genes. The other group are like gas pedal of the car. Our body cells are controlled by these two pedals, gas pedal and brake pedal. Let's start with tumor suppressor genes. As you already know from the previous slide, tumor suppressor genes are a set of genes like P53 and BRK1 and 2. These genes encode for protein responsible for monitoring of the cell cycle and make a decision. If they find any error at any stage, they make one of two decisions. If this error can be repaired, if this error can be solved, then there is no problem. Cell can continue. But if the error cannot be repaired and there are dangers of cancerous transformation, then apoptotic machinery is turned on and cell is killed. So tumor suppressor genes function is to stop cell cycle if there is any problem, provided that this problem can't be solved. Then we have to eliminate this danger. On the other hand, we have another set of genes, like gas pedals, that moves the cell cycle. Genes or proteins helping cell cycle or aiding cellular proliferation, these are normal genes, termed proto-oncogenes. These proto-oncogenes are genes encoding for proteins responsible of moving cell cycle. Like those I told you about, cyclins group, growth factors group of signaling proteins, all genes encoding for these proteins are termed proto-oncogenes. And onco means tumor. So why they are called so? Why are they called proto-oncogenes? It's because any error occurring in these genes results in formation of abnormal protein turning on the cell cycle for 1, 2, 3 times. And instead of stopping at this point, cell continues to divide endlessly. Like a madman driving a car pressing only gas pedal. Cells continue to divide, resulting in terrible outcome. That is why if a mutation occurs in any of these proto-oncogenes, its name changes from proto-oncogenes into just oncogenes. Oncogene means this gene, once a normal gene, but due to a mutation, now it has become bad gene, encoding for abnormal protein, leading the cell to uncontrolled proliferation. And on this condition, This unnatural gene is now termed oncogene. If you want an example, any of the previous genes can be an example, like the genes encoding for cyclins. If a mutation occurs to them, they turn into oncogenes. Genes encoding for signaling proteins like RAS, RAF, and so on. Any mutation of these leads to a tumor and hence called oncogenes. Genes encoding for growth factor or its receptor, any mutation in these genes result in oncogenes. We have finished this concept, so let's shift to this simple photo so I can tell you something about growth factor. But before you forget, I want to test you about something. These growth factors are proteins. May I ask you, which family of proteins do they belong? Are they the group moving the cell cycle or are they the brakes? They are on the proteins moving the cell cycle. They belong to group of proteins turning on cell cycle. Okay? So what are genes encoding for synthesis of these proteins originally named? So what happens if an error occurs in these genes? Growth factors become overactive and cell cycle becomes continuous and gene name changes from proto-oncogenes into oncogene. Now, let me draw your attention to this photo. These are two malignant cells. Molecular biology scientists found that malignant cells, in order to spread and form a tumor, they release these growth factors. Here are some examples of these growth factors. The first one is called insulin growth factor. The second one is fibroblast growth factor, epidermal growth factor, platelet-derived growth factor, vascular endothelial growth factor, and there are so much more. These are just few examples. Cancer cells are continuously releasing these growth factors. Why are they doing so? Because these factors act on specific receptor termed tyrosine kinase receptor. Family of receptors termed tyrosine kinase linked receptors. If any growth factor touches any of these tyrosine kinase receptors, what happens? This receptor, when it becomes active, it sends signals activating specific protein. This specific protein has a name. It can be called Wnt, RAS, RAF, or whatever. So to make it easier for you to remember, let's call it protein kinase number 1. So this kinase is a protein. When this protein is activated, it also activates an adjacent protein. Let's call it protein kinase number 2. It also has a specific name, but don't bother with it. And when this one is activated, it also activates the adjacent protein called protein kinase number 3, then protein kinase number 4, and so on according to the pathway. For example, platelet-derived growth factor has a certain pathway. When it activates its receptor, it leads to activation of a certain number of proteins. This pathway could be 5 proteins, it could be 50, we don't know. Each growth factor has certain pathway, ultimately activating certain protein inside the nucleus, and this protein has a direct relation to the DNA, and this protein activation leads to turning on proliferation machinery in the DNA, so the cell starts to divide endlessly. Okay? This photo I drew for you, or these proteins you see, are called in molecular biology signaling pathway or cell signaling pathway. This concept is somehow advanced and is suitable for postgraduate level. However, I am explaining it for you as an undergraduate because I want you to understand simple point. We can't stop cancer cell until we see it from the inside and understand how does it work. Scientists are working in their labs every day releasing new information for us. So I, a pharmacologist, can design a drug which blocks tyrosine kinase receptor. Now I can do it. If I discovered that this tumor is a result of overactivity of epidermal growth factor, I can block its receptor by designing a drug. If I can't block this receptor, then I can target one of these proteins. I can target protein kinase number 1 and block it for good. I can design a drug for protein kinase 2, 3, 4, 5. All that matters is to cut this malignant chain that can destroy everything and kills the patient. You can intervene with a drug at any level, provided that you know what you are dealing with. That's why people working in labs are extremely specialized. You may find a group of people studying for 20 years only vascular endothelial growth factor. They are specialized in this specific protein, studying everything about it, its pathway, its effects. So once they release these informations, companies concerned with drug design can target whatever they find suitable for their purpose. In the next video, we are going to talk about very recent families of anti-cancer drugs. Why are they called recent? Because once we knew this information, we were able to design drugs which blocks these receptors. We could even design highly specific drugs that can target only one of these proteins, blocking it, thus paralyzing cancer cell. Cancer cell will no longer be able to run its proliferation machinery, so it will stop. Don't you worry about this picture. We won't discuss these complicated concepts. However, I want to draw your attention to one idea that cell cycle is not that simple. This is not internet cables or mobile networks. Each circle you see is a specific protein, and each one of them has a specific name, B38 or whatever. This signaling network you see is the group of proteins controlling cell cycle. Look how much complicated this is. Proteins that has blue lines are concerned with moving the cell cycle. Proteins that has red lines are brakes that stop cell cycle if they notice any error. Look how complicated this is. That's why studying cancer is not so easy and is mainly reserved for postgraduates, because it can be this complicated. Sometimes when we want to make some postgraduate students experience a really hard time, we choose one of these proteins and ask them to follow its pathway. For example, CAK protein. I may ask them to give an account on CAK signaling pathway. Of course, this is such a hard question. Not everyone can deal with it. This is only to give you an idea about how far this can go. In this slide, we will talk about something a little bit new, called telomere and telomerase. This might sound like recent concept, more suitable for postgraduates. However, undergraduates can benefit from it, either in exams or studies. Now I want to tell you about a story called telomere and telomerase. And this is so important for you, especially as undergraduate, because it makes you look so good and confident during exams, especially oral ones. Before we became medical students, we used to draw chromosomes like this. These ends here colored with yellow. If you have a magnifying lens and used it to look at these yellow ends are somehow like bottle caps, covering the ends of chromosomes. This capping part is called telomere. Telo means end and mere means something related to chromosome. This part covering chromosomes is essentially made for two purposes. Both of them are quite reasonable. First one is protective. It protects the ends of chromosomes from erosions, exactly like a cap. Second function is even more important. If there is a gene at the end of chromosomes or at the edges, this gene can never be expressed or dealt with by polymerase enzyme unless it has this extension called telomere. If this gene were on the edge of chromosome without telomere, RNA polymerase enzyme wouldn't be able to read it as if it were dead. RNA polymerase needs something covering the gene so it can read it, translate it, and make a messenger RNA. This can't happen for genes here on the edge without help of telomeres. Those are the two functions of telomere. Anyway, when scientists studied this telomere, they found that this telomere is composed of repeats. 6 nucleotides are repeated, they are called repeats, and they aren't coding for anything. So they don't synthesize any protein, it only has the two mentioned functions. However, I am hiding a third one from you, which we are going to talk about because it is related to cancer directly. I told you about two functions of this telomere. First one is protection or capping. Second one is enabling RNA polymerase to read genes present at the edges. The third function is where we need to draw your attention. Now I'm going to tell you about normal cells, ordinary, non-cancerous cells. If this is a normal cell and is ordered to divide, let's assume that this is a bone marrow cell and body is sending it mitotic signals in order to divide and form a happy family of cells. That is okay. One cell divides into two. Each one of them divides to form 4 cells, another cell cycle, and we have 8 cells. Still, there is something I'm hiding from you. Look at the chromosome of the first cell. That's before any cell division. This is the chromosome and this is telomere. Both are flawless, normal. Their structure don't have any flaw. Once the cell divides, look at the telomere now. It became a little bit shorter. A little part is gone. Third generation. Let's look at their chromosomes. These chromosomes of the third generation are even shorter. With each cell division, something is lost from the telomere, like an erosion. A little part is cut out of it. Fourth generation, look at the telomere. It is nearly gone. As if telomere is getting shorter with each cell division until it disappears. But what could happen if it completely disappears? Cells here are no longer capable of division. Cells say now, thank you, we have reached our limit and we can no longer divide. But why? Because telomere has already gone, as if this telomere is like a counter. With each cell division, telomere gets a little shorter and the counter counts 1. Then the next generation telomere gets more shorter and the counter counts 2. Then 3, 4 and so on till it disappears. In this situation, cells are termed senescent cells. They are now senile. This is what makes our body cells reach a certain point at which it can no longer divide nor function. This gives us white hair as we get older. Wrinkles appear on our faces because fibroblasts used to secrete collagen anytime are no longer able to function because they are now incapable of division. This what makes bone marrow weak at old age, can no longer correct anemias or such diseases. If you want to describe this process with one word, in short, it's aging, because this telomere now, this counter or telomere allows some of your body cells to divide, like 10 times. Some other cells with longer telomeres are allowed to divide more, like 100 times. Others are allowed to divide 500 times. So let's say that telomere length was adjusted according to cell function. If those cells are to divide 500 or 1000 times, then telomere inside the nucleus of these cells are a little longer, so it can withstand cell division 1 to 3 times up to 1000 times. On the other hand, some cells may have very short telomere, so they divide 1 or 2 times and then they say, thank you, that's it. Like cells of the CNS, cells of the heart, kidneys, and so on, they can divide because telomere inside them can't withstand such divisions, because it's short. Why am I telling you all of this story? Because I want you to focus on the concept I am about to explain. What do you think of cancer cells? How many times they can divide? They can divide thousands of times. Then what if they were originally derived from cells that aren't supposed to divide? What makes them go crazy and divide endlessly? It was found out that cancer cells have developed some marvelous strategies, including this weapon called telomerase enzyme. Cancer cells are making such a weird trick. With each cell division, after this telomere gets shorter, this enzyme restores it to its original length, as if it is resetting the counter, so counter starts again from zero. Another division and the second generation is here. Its telomere should be shorter? No, because telomerase fixes the telomere, restoring its original length, so cell can divide once, twice, and so on. This goes on as the counter is now broken. It is resetted after each division. This enables cancer cells to divide thousands of times, so that scientists say cancer cells are immortal. They can't die. Today, we are working on some cancer cells in labs. The origin of these cancer cells is traced back to 70s, so they are 50 years old. For example, a patient had cancer in the 70s and we took sample from these cancer cells and cultured them. To this very day, those cancer cells are pretty much alive. These cells are transferred from one plate to another and they still live and divide. They are immortal. Why are they continuously working? Because they have this telomerase enzyme. Nowadays, this is such a hot topic in research, this telomerase, because scientists are now trying to design drugs targeting this telomerase and disabling it, so cancer cells stop dividing after only 1 or 2 times. This is possible, and it's also possible that we fight aging. This aging process all humans go through. Aren't wrinkles and weakened body result from inability of cells to divide because their telomere is shortened? So let's manipulate this telomere and deliver telomerase enzyme to these cells with any way possible, or making cells capable of synthesizing this enzyme so they become more durable and divide more and more. Anyway, this is now a very hot topic. Every day now scientists are awarded for researches made in this area. However, here in the Middle East, we are not given this area of research the attention it deserves. Finally, after realizing some of the main concepts in this field, we are now able to discuss treatment options. What choices do we have? We have these 5 options. For example, surgery. If I can perform a surgery and remove the tumor, then this is the best option. Or surgery followed by radiation. This can result in better outcomes. Sometimes we can give the 3 options together: surgery, radiotherapy, plus the third one, which is our main concern here, pharmacotherapy. Sometimes we don't know if the tumor managed to spread before the time of surgery, so we need to make sure to eliminate any possible trace of the tumor through sessions of chemotherapy that can last from 6 weeks to 6 months. Don't bother with chemotherapy now, we are going to explain it in detail in the next video. There is a strategy called bone marrow transplantation, especially for leukemia cases. And of course, there is always the palliative therapy for managing the symptoms. Sometimes as you treat cancer patient with anti-cancerous drugs, the patient experiences episodes of vomiting. Treating this vomiting is considered palliative therapy or supportive therapy. This type of therapy is directed at symptoms occurring in cancer patients or what's known as cancer-associated syndromes. We aren't surgeons or radiotherapists. We are pharmacologists, and all that we can do is pharmacotherapy for cancer treatment, and other drugs for treating cancer-associated symptoms such as pain and vomiting and so on. This will be our main topic in the next video. But before I let go of you, I must firstly inject you with a dose of reality. You may dream that since we have anti-cancerous drugs, then tumors should melt or undergo apoptosis and cancer cells die like this. To be honest with you, this scenario could happen and that would be fantastic and we are so lucky. However, man doesn't attain all his heart's desires, for the winds do not blow as the vessels wish. Sometimes after we give a drug and after this drug eliminates as many cancer cells as it can, but for some reasons there are still some resistant cells. They don't respond to the drug. So sometimes you may give a drug initially improving the patient, so you imagine this cured him, neglecting that some of the tumor cells are resistant and don't respond to your drug, resulting in relapse of the patient in 1 or 2 years. Why did the tumor come back? Because in time, those little surviving cells grow again into another secondary tumor. This tumor is more resistant to treat. That's why what happened is like natural selection. When you eliminated all the weak cancer cells, only stronger cells survived, forming this secondary tumor. So, after cancer patients finish their drug sessions and are initially cured, when they come back to us in 5 or 4 years with a secondary or recurrence, this is bad prognosis. We don't expect good outcome. Because we know since this tumor has recurred once again, then it's resistant and this is bad. Because tumor has recurred once again, those cells has recurred after using a drug and those cells are now surely resistant to this drug we used before. That makes our options fewer and this is bad for the patient. You now have a solid base in cancer mechanics, thanks to the main headline we discussed. Not only in cancer, but also in molecular biology. You also have an idea about recent researches in this field. Now you can approach cancer as a specialist, so the next video is going to be so much easier for you.